Inside your body right now, thousands of cells are quietly ending their own lives. This is not a malfunction or disease. This is precisely how your body is designed to work. The scientist who unlocked this mystery transformed our understanding of life itself. Robert Horvitz is a biologist whose curiosity about the smallest creatures led to discoveries that would reshape medicine. Born in 1947 in Chicago, Horvitz developed a fascination with how living things develop from a single cell into complex organisms. This question would guide his entire career. Programmed cell death, also known as apoptosis, is the process by which cells deliberately destroy themselves. Imagine a sculptor chiseling away marble to reveal a statue hidden within. Your body does something similar during development. The spaces between your fingers exist because cells that once filled those gaps received signals to die. Your brain refined its connections by eliminating unnecessary neurons. This controlled destruction is as essential to life as growth itself. Before Horvitz's work, scientists knew that cells could die, but they believed most cell death was accidental, caused by injury or disease. The idea that cells possessed an internal program for self-destruction was revolutionary. It suggested that death at the cellular level was not the opposite of life, but an integral part of it. Horvitz chose an unlikely research partner, a tiny, transparent worm called Caenorhabditis elegans, barely visible to the naked eye. This millimeter-long creature became his window into the fundamental processes of life. The worm's transparency allowed Horvitz and his team to watch every cell in its body as it developed. They could observe cells being born, maturing, and dying, all under the microscope. What made this worm particularly valuable was its consistency. Every healthy worm of this species develops exactly 1,093 cells. During development, precisely 131 of these cells die at predictable times and locations. This reproducibility meant that Horvitz could study cell death as a programmed event rather than a random occurrence. Horvitz began mapping the death of every single cell. He tracked which cells died, when they died, and what happened to their remains. This meticulous work revealed patterns. The deaths were not random, they followed rules. Certain cells always died at specific stages of development. The process was orderly, controlled, and essential. The breakthrough came when Horvitz identified the genes responsible for programmed cell death. He discovered that specific genes acted as executioners, carrying out the death program. Other genes functioned as guardians, deciding which cells should live and which should die. Still others worked as cleanup crews, disposing of the cellular remains. This was a genetic program as precise as any that controlled growth or reproduction. One gene in particular, which Horvitz named CED-3, acted as the central executioner. When this gene was active, the cell would die. When Horvitz created worms with a mutated, non-functional version of this gene, something remarkable happened. The 131 cells that normally died during development survived instead. The worms developed with extra cells, proving that cell death required active genetic instructions. This discovery had profound implications. If programmed cell death was controlled by genes, and if these genes were conserved across species, then understanding them in worms might reveal how cell death worked in humans. Horvitz's hypothesis proved correct. Researchers soon found that human cells possess genes remarkably similar to those Horvitz had identified in worms. The medical significance became clear. Cancer, one of humanity's most devastating diseases, often results from cells that refuse to die when they should. Tumor cells evade the death signals that would normally eliminate damaged or unnecessary cells. Understanding the genetic program of cell death opened new possibilities for cancer treatment. If scientists could reactivate the death program in cancer cells, they might be able to stop tumors from growing. Conversely, some diseases result from too much cell death. In neurodegenerative conditions like Alzheimer's and Parkinson's disease, neurons die when they should survive. In stroke and heart attack, oxygen-deprived cells activate their death programs prematurely. By understanding how these programs work, researchers could potentially develop therapies to prevent unwanted cell death. Horvitz's work also illuminated how the immune system functions. Immune cells must die after fighting infections to prevent them from attacking the body's own tissues. Autoimmune diseases can occur when this process fails. The death program Horvitz discovered plays a role in maintaining the delicate balance of the immune response. Throughout his career, Horvitz emphasized the importance of basic research. He studied worms not because he wanted to cure worm diseases, but because he wanted to understand fundamental biological principles. This approach, sometimes called curiosity-driven research, has repeatedly proven its value. The most practical applications often emerge from investigations into questions that initially seem abstract or irrelevant to human concerns. In 2002, Horvitz received the Nobel Prize in Physiology or Medicine, sharing the honor with Sidney Brenner and John Sulston. The Nobel Committee recognized that their work on programmed cell death in the tiny worm had revealed a fundamental aspect of how all multicellular organisms develop and maintain themselves. Horvitz continued his research at the Massachusetts Institute of Technology, where he has spent most of his career. His laboratory remains active, training new generations of scientists and pushing the boundaries of what we know about cellular life and death. His students and collaborators have gone on to make their own significant contributions to biology and medicine. The elegance of Horvitz's discoveries lies in their universality. The same basic mechanisms that control cell death in a microscopic worm also operate in humans. This conservation across hundreds of millions of years of evolution suggests that programmed cell death is not just important but essential. Life as we know it could not exist without death at the cellular level. Robert Horvitz revealed that death is written into the code of life itself. His exploration of programmed cell death showed that destruction and creation are partners in the dance of development. By watching cells die in a transparent worm, he illuminated processes that shape every human life, from the formation of our bodies before birth to the diseases that challenge us throughout our existence. His work stands as a testament to the power of careful observation, patient experimentation, and the courage to ask fundamental questions about the nature of life.